All right, everyone. Welcome to podcast 4, which is called, as you can see, Introduction to Drosophila. Okay, so what's that? Well, Drosophila is in fact, Drosophila melanogaster is the scientific name for the common fruit fly. Okay, what you're seeing in the picture is what we're going to be working a whole lot with. It is called the wild type fruit fly, which just means this sort of fruit fly has all the normal characteristics we would associate with a wild fruit fly. It doesn't have any weird mutations or any unusual traits that we will see in some other flies that we're going to be working with. In particular, you might want to note, hang on, let me get my pen, there we go. You might want to note the red eye and the wing shape, because these are normal wings. Wild type flies normally have red eyes and these long normal shaped wings, and we'll see some differences from that as we go on in our experiments throughout the year. Okay, so what we are going to do with fruit flies, okay? For our purposes presently, and I'm sure nobody wanted to see that picture, but we are going to be using fruit flies to study courtship behaviors as part of our animal behavior unit. You should have read some information about courtship behaviors and so on, and there's some really ritualized behaviors that flies go through as they're mating. So hopefully we will be able to observe at least some of those. So we'll use them for that initially. But most importantly, we are going to use the fruit flies through a large chunk of the year, at least certainly the first semester, to help with our genetics unit as we look at the results we get when we do crosses with flies that possess different kinds of traits. So that's what we're going to be doing with them. Okay. Why Drosophila? Why not some other organism? And the truth is, you can do genetics experiments with lots of organisms. There are different plants that work really well. There's other things you can do. But Drosophila is really good, especially for genetic studies, for a lot of reasons. Firstly, they're very easy to raise, not difficult at all. They are tiny, which means you can have a lot of them in a small space, which is helpful in a lab situation. They make lots and lots of babies. Lots. And they make those babies very quickly. So even mice that we would think of as having a quick reproductive cycle can't hold a candle to fruit flies. Fruit flies produce tons of young very quickly. Every couple of weeks their life cycle repeats, so you get a lot of babies very, very quickly. And that means, of course, you get quick results for experiments that you're doing. And fruit flies have lots of really easily identifiable traits, so that's very beneficial too. When you're doing a genetic study, it helps that you can identify the traits you're looking at without a whole lot of effort. So that's the why to work with them, why they're a good choice for genetics crosses or whatever you may want to do with them. But the how, okay? Before we do anything, before I even tell you how we're actually going to physically be handling them, I do want you to be prepared that one of the first things you'll be asked to do is simply learn to distinguish the sexes. Okay, and we need to do that whenever if you're doing courtship behavior experiments, you need to know who male and female is, and if you're doing genetics crosses, you need to know which parent is which. So, what we're seeing first here is the male. And what you should know about him, though it's not always easy to tell this when you're looking at them, is he is slightly smaller than the female. The abdomen is darker, okay, and it is more blunt. Now that's hard to tell from this photograph. If you're looking at them from the top down instead of profile, that the shape is more easy to see, but that really heavy, blunt, black butt, if you will, is a real dead giveaway that that's a male. There are sex combs, little bristles on the forelegs of the male that do not show up in this photo well. This is obviously the easier characteristic. If this is for some question in doubt, you can look for little hairs sticking off the forelegs, but hopefully the black bottom will be very evident. And of course the female is just the opposite of all those things. And her abdomen sometimes can really deceptively look dark at the bottom. This is very clearly striped, but if you see a male and then look again, you can any lightness at all in the end, you've got a female. That male is always very heavily dark on the back. So even if her stripes are kind of fusing together a little bit, usually you can still tell the difference. Now, when we're working with Drosophila, we need to understand the life cycle. The first thing we should know, here's your male and female flies right here. When they mate, the female is fertilized internally. All right, she will carry sperm from her very first mating inside her body for the rest of her life, and her eggs will be most likely fertilized with that sperm, even if she mates with other males over the course of her very short life. That said, once her eggs are fertilized, she will eventually lay tiny little embryos, which would be, reasonably difficult to see. But those little embryos will start growing into larval stages, which, much like a caterpillar is to a butterfly, it's a feeding stage. And the vials that we have in class, and you'll see them shortly, there is a blue gooey substance in the bottom of the culture vials, the containers that the flies are in, and this blue stuff is simply their food, and these little caterpillary type larvae are just burrowing in that food, and they slowly grow into different stages, and finally the last stage of this caterpillary type then gives way to something resembling a pupa, or a cocoon stage, if you will. This pupal stage is the last stage before immediately hatching and the adult emerges, and then you begin the cycle all over again. Again, from egg to adult, we're talking about 2 weeks, and that will vary depending on your conditions, particularly temperature. If it's chilly, it will take longer. If it's warm, it usually goes quicker, provided you don't cook them, in which case that generally is not good. Okay? Now, this life cycle has real implications for how we do any kind of experiments we're doing. As I mentioned, fertilization is internal, and the females store sperm for life. Okay? That means if you are a male fruit fly, and please remember, male fruit flies are not thinking about this, but their behavior has been modified by natural selection over time, such that males behave such that almost as though they understood that it is beneficial to them to be the first one to fertilize the eggs. In other words, it's best to mate with a virgin female as opposed to one that's already mated, because his sperm don't stand as good a chance of getting to those eggs. So that's an important thing to think about with our courtship experiments. And then for the nuts and bolts of actually doing our handling of the flies, when a female emerges from that cocoon, that pupal state, she's not receptive to males yet. She has to mature. That takes about 8 hours. So 8 hours after a female fly emerges from a pupa, she will be ready to mate. And if you do not remove her from a vial of flies where there happen to be other males, she will mate with those males. And then if you pull her out, she's already storing sperm. And if you wanted her to mate with someone else, well, it's too late for that, because the sperm that she's got are whoever was in the vial with her, and so you blew it. Bottom line is, we have to obtain virgin females for our crosses that have never mated before, and that means that we have to be ready to essentially pull those newly hatched females out of a vial and put them into a clean vial by themselves immediately. We can't let them sit in there for longer than 8 hours, or we run the risk of them having mated with whatever males might happen to be in the culture vial with them. Okay, so timing is really critical, and we're going to have to be vigilant in order to get these flies and have them in the state that we need them to be in. Okay, so we want to be able to control parentage and so on, so we've got to remove those females before 8 hours has passed from the vial. All right. Again, why do we need these virgin females? Well, for our courtship experiments, since it's so important for males to be the first to fertilize, it may very well be that they respond differently to a female that is a virgin versus one that has already been fertilized. Then again, maybe they don't, but maybe that's an experiment we could run. But suffice it to say that it might likely be an affecting factor. So we want to do this with virgin females, so we'll see the best courtship displays possible. And of course, for genetics experiments, if you don't know what your parent, or if your parentage is not correct, or if there's possibility of more than one parent, then your experimental results go out the window. Okay, so we got to know with certainty who our parents are. Okay. So the first thing that we will do is we will work with our flies, existing culture of wild type flies, just to practice telling males and females apart. Okay, and when we do that, you have to learn how to make it so they won't fly away when you're trying to observe them, and that means that you have to be able to put them to sleep. There is a little drug that we use to do that, and it's called fly nap. And in a video that follows, I'm going to show you how to do that. Okay. Additionally, we are going to have, once you have observed flies for male or female from a culture vial that I give you, okay, we are going to have to basically not return the adults back into that culture vial, but leave that culture vial so that it contains only the pupa and the larva. And we're going to have to monitor it closely, waiting for the virgin females to emerge, and we'll talk about that as well. Now, so that means once we have observed male and female out of our culture, we're not going to return the adults back into the vial. We are going to have to kill them. Sadly, that is what is done. There's fruit fly mass murder going on every day in genetics labs because this is how you have to handle them. So, once we get rid of the adults, you'll have a vial that is just these larval and pupal offspring, and someone's going to have to come back after school from your lab group and check for any females that might have emerged. Okay? It doesn't have to be the same person every time, but somebody's going to have to come in, sign in, and check for those virgin females. That will be part of your group's lab grade. So you work it however you want. I don't care who comes, but somebody needs to come. You can split it up. You can be the same person. You all need to agree on it. But if your name hasn't been signed in and someone hasn't come and checked them, then you will get a deduction from your lab grade. Okay? And then we'll have a little section later on learning how to recognize different traits. That's all that last little slide was about. See? Okay, that's it. I'm going to show you a little video showing you how to work with the flies, and I will see you guys later. Thanks for watching.
New fruit fly commercial. Take one.
Thanks, Grace. Okay, the purpose of this little clip is to show you guys how to work with the fruit flies. You would get a culture that looks like this. It's labeled with what's in here and the date that it was put in. The blue stuff at the bottom is their food. It is just starch with some stuff in there to prevent fungus from growing, but if they fall into it, when they are disabled, which we're going to do to them in a minute, they can't get out of it. So it's really important that we not let them fall into that blue gooey stuff. Okay? Now, these are normal, what are called wild type flies. These have all the normal characteristics of fruit flies, and that's what we'll be working with first. And before we can work with fruit flies, the first thing we have to do is we have to put them to sleep. So there is a chemical, and you all will work with this. It's called fly nap. Okay? And what you do is you put the little wand, dip it in the fly nap, okay, and what I would do is pull it up like this so it's not hanging down. Now, this is where you have to be a little bit skilled. You need to tap the flies down, and you got to be quick. Tap them down, and then whoop, and see, I had a few escape on me. Now, what you want to do is lay the vial down on its side, because if you keep it up straight, they're going to fall down into the blue goo when they fall asleep, and they will they'll die in that goo. So what we're going to do is let them fall asleep with the vial sitting on its side, because that way they'll fall onto the plastic in the vial and we can get them out much more easily. Okay? So I'm going to set that aside for a minute, and while we're doing that, I want you to see how you set up a new culture vial, because sometimes we want to put some of the flies that we've put to sleep in here into a new culture. So I'll show you how to do that. The first thing you need is some food. The food comes at first in this dry powder form, and you just get a scoop and you pour it in. Okay? And then you're going to get water in it, and sometimes if you look at the bottom, sometimes it's hard to get the bottom thoroughly wet, so you kind of want to jet the water in there a little bit so that it sinks down into the bottom layers of the powder. Okay, and the idea is that you get it wet without getting it completely soupy. Okay, so you want it to be completely blue, but you don't want it to be so liquid that it's running around. So that kind of sometimes takes some getting used to. Like if we look in there, I think that probably needs just a little bit more water, because I'm still seeing dry flakes, but if I add too much, it's going to get too wet. Okay, so, and see if you can do that. It looks all blue, but you hold it upside down and you don't get, that's probably about right. I might add just a little bit more in there. Okay, so, and then you would have a cap ready to go. Now, I'm looking at these flies, and what have I been talking? Maybe about a minute, 2 minutes, and they are already starting to fall asleep.
Wow, what did you do to them?
I gave them a little anesthesia. Okay, and pretty soon, I'll give them just a few more seconds, we'll be able to put them in a petri dish, and that's what you'll want to put them in so that you can observe them under the microscope and determine males and females and so on. So I think they look like they're about ready, so I'm going to gently remove the stopper. Okay. And I'm going to try to flip these guys into this petri dish. Okay. And if you've got some stubborn ones clinging to the sides of the wall of the vial, sorry, I should have had this on the table, you can get a little paintbrush and kind of brush them out with that so they're not in there. Now, I really, really want to get all of these adult flies out of here, and the reason I do is because what we are going to need to do all of our experiments are virgin females, and we talked about that in the PowerPoint. You need females that have never mated before, so that they aren't storing sperm, so that way we have control of who mates with them. So I think I've got all the adults out of here. So what we would do with this vial now is we would wait, and we would check this, and it's very important that once 8 hours passes from this point, we come in and check it to see if there's any females in there, because if we go much past 8 hours, if there are any males that also emerge from these pupa in here, then they will likely mate with each other. There's only about 8 hours from when the females emerge that they are not receptive to males. After 8 hours, they could be mating in there, and that won't do us any good. So we've got to come back and check this, and this is part of your lab grade, that you will come back and check your vials regularly for females. Now, as far as this, if we wanted to look at these flies just to study them, to learn to tell sexes or whatever we might want to do, you can put these under a dissecting microscope using your paintbrush, move them around as you need. And what? I'm sorry, my daughter's filming this for me, and she's sending me secret messages. At any rate, you can observe them, and when you're done with them, assuming you don't need them for some purpose, you might need to cross them with something, but since these are not virgin females, we wouldn't want to cross them. You do, generally speaking, kill laboratory fruit flies, and the way you do that is you drop them in alcohol. Each group will have an alcohol vial, and you will just put any unneeded flies into the vial and that's also called putting them in the morgue, and they are killed by doing that. Okay?
You're dropping stuff.
I'm dropping stuff. And so I will clean this up. You don't need to watch that, but that's pretty much everything you need to know, at least to get started with working with the fruit flies. Okay? And we will talk about how we switch. Sometimes if we had a female emerge in here and we want to put her in a new vial, then that's a little tricky how we do that, so we'll talk about that. But I think you pretty much got it. Thank you very much. Thank you, Grace, for filming, and I'll see you guys soon. Bye.